Good morning, everyone. My name's Angus Brown, and in my role as the incoming chair of the Physiological Society's History and Archives group, it's my pleasure to introduce Professor Semir Zeki, who is giving the annual Paton Prize Lecture. And this lecture originated from a donation from Sir William Paton, the famous English physiologist, pharmacologist, and Fellow of the Royal Society. And his extremely important contributions to physiology and medicine was the discovery of two classes of drugs that acted at the nicotinic receptor as antagonists. Decamethonium, which was the first specific neuromuscular blocking drug, which was a muscle relaxant and was used to induce paralysis during surgery, and hexamethonium, which was used to lower blood pressure. And today's speaker is Professor Semir Zeki, Professor of Neuroaesthetics at UCL. Professor Zeki's work is the study of the visual brain, with his recent work focusing on the relationship between visual art and the functioning of the visual brain. So it's my pleasure to introduce Professor Zeki's talk, the title of which is A Colourful Experience.
Thank you very much, Chairman. I am greatly honored by being invited by the Physiological Society to give this Paton Lecture. William Paton, being a man whom I actually knew and much admired, was a great supporter of physiological and pharmacological sciences in his position as a trustee of the Wellcome Trust. Now, I'm going to be talking about one small area of my work, which is my work in color vision. And I'm going to put it in a somewhat historical context and give you my colorful experience of my work in that area. When I first started work in the visual brain, which was in the 1970s, early 1970s, the general view we had of the brain was that it was the primary visual cortex here, which is shown at the back of the brain, which receives, it was thought, the only recipient of visual signals from the retina, the only direct recipient of visual signals from the retina, known as area 17, V1, or the primary visual cortex. And the most complete picture of that was given by David Hubel and Thorsten Wiesel, who had shown that the cells of the visual brain do not respond to light as such, as generations of scientists had thought, but that they respond to lines of specific orientation, or rather transformations in energy between one part of the receptive field and another, which took the shape of oriented lines. Now, these oriented lines were arranged in the cortex in a very, very orderly way. Surprisingly, in the summary of their work delivered which was delivered in the Ferrier lecture, which was delivered in 1972 but published in 1977, they wrote that in the primary visual cortex, or area 17, color information is not taken into account at all. It was therefore surprising to me and to many others when exploring the areas of the brain outside the primary visual cortex, in these regions here, what was known as the visual association cortex, I discovered a region which was, this was in the monkey, and done electrophysiologically. I discovered a region which had very heavy concentrations of what we might call color code cells. I will specify the term later much better. This was surprising because this is an area of the brain which is not directly recipient or thought of signals from the retina, and it was an unexpected discovery. The position of the area I called V4, this color-rich area, and its position in the human brain is seen in this slide. It is located ventrally in the brain in a region called the fusiform gyrus, and it consists of two areas, V4 itself and V4 alpha. So V4 is very heavily color concentrated, and V4 alpha also has color, but is also concerned with form. So we refer to the whole area as the V4 complex, and so it is concerned with color for V4 and color in association with form for V4 alpha. This is, of course, done, the first demonstration of this concentration of color cells was done in the monkey, and this is the picture which we obtained from the human brain. Now, more recently, about 4 years ago, Bevel Conway and Lafayette Souza and Canwisher in the United States have in fact shown that this area, which the area V4, which is here shown in orange, is actually sandwiched between areas that are responsible or are critical for the perception of faces here and perception of places here. There are lots of specializations in the brain, and the color region is sandwiched between these two. This is a region which is actually indifferent to form and concerned with color. This is the most recent definition of, it doesn't vary very much, except it shows that the area is sandwiched between two other specialized areas. Now, damage to this area causes the syndrome of cerebral achromatopsia, or an incapacity to see the world in color. Such subjects only see the world in black and white. The syndrome of cerebral achromatopsia had been described before, but it had been thought that it was due to hysterical states, not to organic damage in the brain. In fact, I'll show you what the syndrome consists of. If you have damage to area V4 of one side, then the opposite field of view appears in shades of gray. And usually this is described as dirty shades of gray, like this. And this is due to damage to this area here, area V4. Now, so to summarize then, there is an area in the brain which is in which there is a heavy concentration of cells dealing with color, and that it is damage to it leads specifically to the syndrome of cerebral achromatopsia, or an incapacity to see the world in color. Such subjects are able to see the forms, to see the directions of motion, to see distances, and to read, but they are not able to see the world in color. So to all intents and purposes, it appears as though it is a specialized area. And in fact, you can show this functional specialization in the brain, which I think is a hallmark of the organization of the visual brain, by this one slide, which in fact summarizes the whole organization according to functional specialization of the brain. When you get subjects to look at a pattern of a colored pattern of abstract colored pattern with no recognizable object, you have activity in area V4 here, right? And when you get the same subjects look at the pattern of black and white dots in motion, you have activity in a very distinct area, area V5. Both of them, of course, activate, both stimuli activate the primary visual cortex, area V1, which was and still is really the royal entrance to the rest of the visual brain. So area V1 acts as a distributor of signals to these various specialized areas for further and specialized processing. Now, specialization is, I think, one of the fundamental principles along which the visual brain is organized, and many other specializations have since been discovered since I published a review in 1978 on functional specialization. There have been specializations discovered for faces, for facial expressions, for bodies, and for places, and other things besides. So the question is, why do we have functional specialization? And there are several reasons, at least two reasons for it. One is that events in the visual world do not co-occur. In other words, a red bus can be moving to the right, but the color red can invest a bus moving in any direction. Indeed, it can invest any object, and any object can move in the right direction. So if only a red bus could move to the right at a given speed, then the speed, the direction, or the color and the form could uniquely specify that. But it is not the case. These things occur independently of each other in the world. The other reason is that the requirement, as I will show you presently, requirements for constructing colors in the cortex. Now, I do use the word constructing rather than representing advisedly. The requirements for constructing colors in the cortex are very specific and different from the requirements for constructing, say, motion. For color, you have to look at the wavelength composition. I'll explain all of this in a moment, but let me just finish with the functional specialization. For color, you have to gauge the wavelength composition of light coming from one area and from surrounding areas simultaneously in time. For motion, you have to integrate information coming from at least two successive points, two different points successively in time. So these are pretty good, compelling reasons for why you should have functional specialization in the brain. And this gives you an idea of it. It's the Eiffel Tower, which we can all recognize very soon after Bastille Day. The Eiffel Tower can be invested with any color. In the center, it was in the tricolor for the Bastille Day. On the left is orange, and on the right you see it as it appears in daylight. And yet it does not lose its form. So the form is independent of the color, and the color is independent of form, because the blue could also invest any other object. Hence, let me specify again why we need functional specialization. We need it because events do not co-occur, and because to process different attributes of the visual scene, for example, motion and color, the requirements, at least the temporal requirements, are very different. So the functional specialization, which you see in terms of anatomy and physiology, because these are geographically different areas with different properties, that functional specialization is projected in time. So if you ask subjects to, for example, in this example, this is a very simplified example of an experiment which I did with Konstantinos Moutousis, in which a pendulum swings back and forth, all right, and changes its color as it swings back and forth. But you can do the experiment with a square moving up and down, changing its color as it moves up and down, or it could be many squares moving up and down. There's a number of ways of doing the experiment, but essentially the experiment is that of an object that moves in one of two directions and changes its color from one color to another. And the task for subjects, let me just say, the change in color and the change in direction of motion is in and out of phase. And it's a bit faster than what is shown here. You can also do it in this way, by looking at these squares in a complex scene. So the squares can be either red or green. They're illustrated as red and green here, but they can be either red or green, and they can be moving either up or down. So the task for the subject is to say what color the stimulus was when it was moving up, all right? Or what color it was when it was moving down, or what was the direction of motion when it was red. And if you ask subjects this question, you'll find that they will perceive consciously, because they have to report the color of the stimulus and the direction of motion, you will find that they perceive color some 80 to 100 milliseconds before motion. And if we can do this also with form, with oriented lines, you'd find that they would perceive the color some 40 milliseconds before they perceive the orientation. In other words, there is a specialization in time. Now, 100 milliseconds is, of course, a joke. It's a tiny fraction of time, but in physiological terms, it is a huge, huge amount of time, because it takes up to 1 millisecond for the nervous impulse to cross from one cell to the other. Therefore, what we can say is that since you become conscious of color before you become conscious of motion, and you can become conscious of color before you can become conscious of orientation or form, that the visual consciousness is distributed in space and in time. It is distributed in space because visual consciousness for color is the result of activity in area V4. That for motion is a result of activity in area V5. And for form, it's in V3 and V3A, V3B. And it's distributed in time because you see these attributes at separate times. So the notion that there is unity of consciousness is not correct at this early phenomenal level. It may be at the higher levels of the level I am aware of being aware, or I'm the source of the awareness, but it is certainly not true at the level of at the phenomenal level. There is a disunity of consciousness. You become conscious of different attributes at different times. Moreover, that there is a disjunction between the veridical reality and the perceived reality, because these two attributes are presented simultaneously but are perceived differently. And the third point is, a very important point, that there is no part of the brain that seems to wait for all these areas to complete their processing. The brain binds what has been processed. So because color is at time 0 color is processed before motion, so the color at time 0 is combined with the motion that was perceived 80 milliseconds before. So there is no brain area that's waiting for all these areas to complete their processing. Hence, the functional specialization, which involves color and motion and form and many other attributes, is a very cardinal key to understanding the organization of the visual brain. Now, when I first described the color center in 1972 at St. Catherine's College in Oxford, someone in the audience said to me that I had discovered the philosopher's stone. This comment surprised me some. I took it as a compliment, but I did not really understand it because I'm not really very philosophically inclined and had not really read much philosophy at the time. And yet, when one thinks about it, it is a very important area in the study of philosophy. And many philosophers have addressed the question of color vision, but none has done with much merit. The reason why color is so interesting from the point of view of philosophy is that color is a means of obtaining knowledge about a certain property of objects and surfaces. And knowledge, how we acquire it, and how sure we are of what we know, is one of the central problems, if not the central problem, of philosophy. So this is summarized in Western philosophy by the Heraclitean doctrine of flux, the statement of Heraclitus that one never swims in the same river twice, which was taken up by Plato and made almost the basis of his philosophy. And in the Eastern wisdom is summarized as the very common saying in the East that nothing is permanent except change. So the problem with color is to understand how colors remain constant in spite of the ever-changing information reaching us from the surfaces and objects all the time. Now, at the time I started this, as I say, I was not at all a philosopher, and the discovery of a color center came as a surprise, and therefore I was trying to lean on people with much greater experience than me in color vision for their advice. Now, the advice was unanimously given with the best intent, but all of it was irrelevant. All of it was irrelevant because of the habit at the time of not separating sensation from perception. I was told to go and learn colorimetry and various other things and learn about macadam ellipses, all of which were useless for me. And I think there is a lesson in that to be learned, which I'll come to later. But the reason for the uselessness of the very well-meant advice that I was given was that the advice itself was based on a way of thinking which was not appropriate for the cerebral cortex, although it had been highly successful in studying the retina. And it has its roots in the discovery by Isaac Newton that white light could be passed through a prism and broken up into its components. Now, the fact that white light could be broken up into its components made of this a great experiment in physics, and the fact that the components were perceived to differ in color made of it a great experiment in psychophysics and perception. And Newton himself was quick to draw the conclusion And he wrote, every body reflects the rays of its own color more copiously than the rest, and from their excess and predominance in the reflected light has its color. In other words, the green surface looks green because it reflects more green light, and the red surface looks red because it reflects more red light. This statement is actually not entirely true, but there is another statement which is not often quoted, made in the same book, Optics, published in 1704, which is of great importance, in which Newton said, for the rays to speak properly have no color. In them there is nothing else than a certain power and disposition to stir up a sensation of this color or that. And I took it as my task to learn whether the stirring up of a sensation of color occurred in the area that I had discovered. Now, the implication of what Newton was saying was that if you look at a surface such as this one, small area of the field of view, the color of that field of view will be determined by the excess of the wavelengths of light reflected from it. That will be registered by the retina through the receptors, and registered then by a point in the cortex defined as an area 1 millimeter square. So by registering the color of every point through the wavelengths, through the dominant wavelengths that the surface reflects, the brain can build up a color of the entire world. That was the thinking that was prevalent at the time, and actually is still prevalent in many, many places today. Now, Thomas Young qualified this in one of the great insights in sensory physiology, not just in color vision. When he gave his Bakerian lecture to the Royal Society in 1801, he wrote, and I quote, now, as it is almost impossible to conceive each sensitive point of the retina to contain an infinite number of particles, each capable of vibrating in perfect unison with every possible undulation, it becomes necessary to suppose the number limited, for instance, to the three principal colors, red, yellow, and blue. Now, two things to notice about that. One, that he's equating the color with wavelengths. Two, is that he's making a statement which is not only a great statement in color vision, but a great statement in sensory physiology, because what he is really saying is that the brain cannot equip its possessor with a receptor for every single experience that it's going to encounter, because it does not know what experiences the individual will encounter. So it endows the receptors with such potential that they can acquire as many experiences as possible. And Hermann von Helmholtz, the great German physicist and psychophysicist, in fact, drew curves for how you would expect these three curves to respond, these three receptors to respond. He wrote, and I quote, a red light stimulates red sensitive fibers strongly and the other two weakly, giving the sensation red. Notice he's equating wavelengths with the perception of color. Green light stimulates the green sensitive fibers strongly and the other two weakly, giving the sensation green. And when all three fibers are stimulated about equally, the sensation is that of pale or white or pale hues. This is a quote from his book. So this theory of the limited receptors, limited to three, became known as the Young-Helmholtz theory. And when in 1962, Marx, Dobell, and McNichol in the United States isolated the cones in the primate retina and found, when studied their absorption for light of different waveband, they found that indeed there were only three types of cones. I think John Mullen in Cambridge would probably say there's a fourth one in women with for the long wave. So that three, maybe four, but limited number anyway. But one thing that you observe about these, the absorption spectra of these cones, is that they're extremely broad. They span much of the visible spectrum, although they absorb most efficiently at a given point. But if you take, say, the middle wave cone, you can make this middle wave cone absorb light at the peak sensitivity of the long wave cone by simply increasing the intensity of the light. But once a cone absorbs a photon of light, it is no longer able to distinguish the provenance, which part of the spectrum it came from. And this is known as William Rushton's principle of univariance, William Rushton being one of the most astute people who worked on color vision in the world. Now, what was missing in all of this was constant color categorization or color constancy. The word color constancy, you will find it very difficult to find a mention of it in textbooks of physiology or in psychology before 1972. Although Helmholtz and Hering had both referred to it, it was not something that it was sort of relegated to a footnote, if at all. There was only one man who had made a big issue of it, and that was the man who came to see me in London in 1978, and he was one of the great inventive geniuses in America in the tradition of Edison, and his name was Edwin Land, the originator of the polarizing sheet. And since most of you perhaps have not seen him, I thought I'd show you a brief clip of what he looked like.
I want to show first an experiment which we did in 1955, which seemed, on the one hand, very startling, on the other hand, extremely simple.
So when Land came to see me, the discussion became very animated and very excited, and it was, I think, one of the most exciting days I have ever spent in the lab. We went in the afternoon to buy various math papers to reconstruct as well as we could the experiment in the lab. He came with his assistant, John McCann, himself a very good worker in the field of color vision. And he convinced me of the validity of his approach to studying the responses of cells in the cortex. And I was more convinced of him. He was the unique person who convinced me that he had an approach that would be valid and fruitful for studying the cells in the cerebral cortex. And his experiment essentially is as follows. So you have an audience, such as yourselves, and a multicolored display here, which is lit by 3 projectors, one passing long, one middle, and one shortwave light, or if you want to translate into in approximate terms, red, green, and blue light. And the intensity of light coming from each projector can be varied by means of these rheostats and measured by means of a telephotometer. Now, the audience, the audience is asked to look at a nominated square, for example, this green rectangle, and this green rectangle is made to reflect, in the first experiment, 30 units of long wave light here, 30 milliwatts per steradian per meter square, but I'll call it units of long wave light, 60 units of middle wave light, and 10 units of short wave light. And when all 3 projectors are switched on, the audience reports this area to be green, which of course would not be surprising to anyone because it's reflecting a lot more of the middle wave or green light. And this, of course, somehow supports Newton's statement that everybody reflects the rays of its own color more copiously than the rest. The second part, and that from the excess and predominance in the reflected light has its color, is belied by the second part of the experiment, in which you go back to the same green rectangle and arrange it such that this area now is reflecting 60 units of long wave or red light, 30 units of green light, and 10 units of blue light. And when you switch all 3 projectors on, the audience reports this area to be green. So it's a slightly darker shade of green, but belongs to category green, even though it's reflecting twice the amount of red light. Now, if you look to see what happens when this area is reflecting 30 units of red light, the surround, defined as 10 degrees in all directions from the borders of this green area, reflects more red light because it's got a higher efficiency for reflecting red light, and there is a ratio in the amount of red light reflected from this pattern from the surround, which is 0.75. But the same ratio obtains when this area is made to reflect 60 units of red light. The surround is still more efficient in reflecting red light, and therefore reflects more. And there is a ratio of 0.75, which is identical to the one obtained in the first experiment. And the same is true for the other two ratios. Now, there is no physical law which says that these ratios should be taken. It is a brain law, and the result of that brain law is the determination of color, as I shall show you at the moment. So let's go. Next point then is to ask ourselves, where is the cortical site of the ratio-taking mechanism for the construction of colors? Notice I'm now saying the construction of colors, not the representation of colors, which is, I dislike it because it implies representation of wavelength, the construction of colors by the cortex. And you can study that by taking a multicolored patch, which is simulated to have the wavelength composition changing all the time, as you would when you're walking down the street on a partly cloudy day, but the colors remain the same. For example, this blue remains blue and the mauve remains mauve. And you look at the activity in the brain when subjects look at a pattern like this and compare that activity to the activity produced by looking at its achromatic counterpart. And when you do that, you find the activity occurs in area V4, this time seen ventrally. So to the best approximation today, the ratio-taking mechanisms that generate color are located in area V4. Now, please don't misunderstand me. I'm not saying that area V4 acting in isolation does this. I mean, if you put V4 in a Petri dish, it's not going to see colors. Of course, it depends upon pathways that feed it and, of course, the output from it, but it is a central area for working out these ratios. Now, trichromacy, as I say, is it's difficult to understand the color constancy in terms of trichromacy, but people have relied a bit, well, quite a bit, on perceptual opponency, namely that there are, and perceptual opponency has been described as being decided by opponent connections between the three receptors. So here are the very, very broad absorption spectra of the cones, but if you then oppose the red, oppose the green, then you've got the long wave and middle wave opposing short wave to give you yellow and so on. Then you get cells with much narrower action spectra. In other words, they respond to much narrower parts of the visible spectrum. And these then could perhaps be explained better color opponents, the generation of color. And this was, of course, a great, very interesting discovery made by Svitichin and then by Russ DeValor in the United States. And if you concentrate here at this cross for a second, and then look at the white screen, you will see the after images. But and this after the opponent color system was studied in great detail and quantified by Herbert and Jameson in the United States. But the relationship between the unique hues, the hue which is neither red nor green, neither yellow nor blue, and the action spectra of the cells is very loose. And therefore, it is not easy to say that this is really the basis of the color constancy. Although people have tried to do that and have specified that it is the adaptation. So if you look at the green surface, the middle wave cones adapt, leaving the stage to the long wave or red cone, therefore you see red. I don't believe that. And I don't believe it for the simple reason that I have done an experiment, which I am very surprised that Land did not do, and I'm very surprised that I did not do it for a long time. In fact, this experiment was only done in the past 3 years. And what I did do was to get a multicolored display with my colleagues, Dimitris Milonas and Joshua Pepper, get a multicolored display and let subjects look at these patches, all of which were reflecting the same or nearly identical triplet of energies of long, middle, and short wave light. Now, here is a patch that's reflecting more red light than green light. Now, the after image of that, of course, by the adaptation theory, by the opponent theory, should be green, but actually it is red. So the color of the after image depends not on the wavelength composition of light reflected from that surface, but on the perceived color. And the perceived color is generated before the opponent color is generated, and therefore the opponent color cannot explain the generation of the color itself. And if you look at the charts there, whenever red, the frequency with which subjects report that the after image of a green surface is red is 90%. Here it's 100%. It's 100%. With the blue, it is a bit lower. So with many people undecided. So the color of the after image does not depend upon the wavelength composition of the light reflected from it, just as the color of a patch does not depend upon the wavelength of the light reflected from it alone, but also depends upon light reflected from the surround. And therefore, the opponent color is generated after colors are generated, not before. And therefore, they cannot explain the construction of colors in the cerebral cortex. Now, so I have mentioned this, and I'll just pass this by. Now, I want to tell you that if you go to these broad absorption spectra of the cones, I measured the action spectra of cells in the cortex, and you find they're distributed all along the spectrum, and there are some very narrow ones amongst them and some rather broad ones. Land himself assumed that there are 3 channels. I suppose he was impressed by the trichromatic theory. I see no reason to restrict ourselves to 3 channels. There could be many, many channels which are inputting into the color system in V4. And by the way, many people have now shown that the cells in V4 are much more, their properties are much more related to perceived colors than to the wavelength composition of light. Now, let's go back to the brain and the philosophy. Major function of the brain is to obtain knowledge. And to do that, the brain must stabilize the world so that it extracts information about its constant features. And color vision happens to be one of the best examples of how the brain stabilizes the world to extract its constant features. The question to ask is, while stabilizing the world for color, what information does the brain discard and what does it utilize? And it's quite obvious that it discards all the wavelength, all the changes in wavelength composition. These are unnecessary, well, they are important, but they do not specify the color of a surface, because the colors of surfaces remain constant in spite of changes in wavelength composition. So what we have to do is to separate wavelengths from colors. This has been, color wavelengths are very important in color vision, but they can nevertheless be separated. And you can do this by looking at the electrophysiology of single cells, which I did this years ago. So if you take a cell in V1, this is recording from the primary visual cortex, and you plot its action spectrum, so it responds only to long wave light, which in isolation looks red. So it's normal to assume that it is a red cell. But when you put these different areas of the multicolor display in the receptive field of the cell, the red area, the blue area, the green area, and the white area, and make them reflect the same triplet of energies, in this case 69 and 30 and 10, you find that it will respond to each one of these areas with equal vigor. In other words, the cell is colorblind. It's only interested that there should be enough long wave light in the light reflected from the area in its receptive field. Because when you switch the long wave light off, the cell ceases to respond. And if you take an on and off cell, the opponent cell, the cell response gives you an on response to the short end of the spectrum and an off response to the long end of the spectrum. Nevertheless, it can be made to give you an on or an off response to an area of any color. Regardless of its color, depending only upon how you manipulate the amount of long, middle, and shortwave light reflected from that surface. So this opponent cell, which was supposed to be the basis of the perceptual opponency, is not actually, cannot actually be the direct basis of the perceptual opponency. This opponent cell is found in V1. But on the other hand, when you go to V4, you find here a cell that also responds long wave end of the spectrum only, just like the cell in V1 I showed you a moment ago. So you can assume it's a red cell. And when you put the different colored areas in the receptive field of the cell, you find that this one responds to red only. And this is a genuine color-coded cell, which is different from the wavelength selective cells that I've just shown you. And because most of you perhaps have not seen what a genuine color-coded cell is, I've retrieved a recording which was made in the 1980s, and I will play it for you now. Does not respond to white light. Better to red light. But when you put a red area of the multicolored display in its receptive field and illuminate it with long wave light alone, it doesn't appear more reddish. There's a red wash everywhere, and the cell does not respond very well, as you will see now. It's only when you add the other two that the area becomes completely red and the cell responds well. So it does not respond to this illumination, but now it will respond. And so I hope I have convinced you of the responses of the cell, not to wavelengths, but to colors. This is not to say that wavelengths are not very important for color vision, but they can be separated out at the cortical level. And this is something I published in 1983 in a paper called Color Coding in the Cerebral Cortex, the Reaction of Cells in the Monkey Visual Cortex to Wavelengths and Colors. And so now we can address the question, what is discarded and what is not? What is discarded is the changes in wavelength composition. What is not discarded is the reflectance of surfaces for light of different wavebands, because that never changes. So this surface, for example, which is a red surface, will reflect 90% of the red light, long wave light incident on it, no matter what the quantity. So in this case, it is reflecting 90 milliwatts, 90%. In this case, reflecting 270 milliwatts, but still 90%. And you can just see that when you've got various surfaces juxtaposed, their reflectance remains the same, and therefore the ratios remain the same, right? As I told you in this previous demonstration. So I think we can say that color is a construction of the cerebral cortex. It's constructed by the cerebral cortex from the wavelength of light, but it's constructed by discarding the changes in wavelength composition and concentrating on the reflectance of adjacent surfaces to the same waveband by taking the ratios. These ratios are taken by the brain. There is no physical law why they should be taken. And the power and disposition to construct colors resides, as far as we can tell so far, in V4, but V4 does not act alone, of course. It depends upon the input to it. I am interested that in 2020, Stephen Chevelle and his colleagues published a paper in PNAS, which starts with, there is no color in light, color is in the perceiver, not the physical stimulus. I'm interested in that. I'm very pleased about it to begin with, but I'm very interested in that because the paper that I tried to publish, I can't remember it. which I just showed you, was actually rejected by the Journal of Physiology in a very vicious way. It was rejected with a 30-page diatribe against that paper. And to the enormous credit of the Physiological Society, and due in no small part to the pressure given by David Whitridge, a man described as a man with a high IQ and low pH, he complained to the editorial board, and the man who had been dealing with that manuscript was asked to resign from the editorial board. This was not the only reason, I'm told, why he was asked to resign, but this is one of the reasons. I'm also pleased because when I first presented these results at the Physiological Society, it was not easy going. Someone got up and said that you're pretending it's all done by magic and that you're making propaganda for the color constancy boys. And that the record of that meeting is perhaps worth seeing. It is in the archives of the Physiological Society. So finally to return to the philosophical point. Immanuel Kant, who was one of the greatest philosophers of all time, said that we cannot know the thing in itself, das Ding an sich, because our knowledge of the world is interposed through the mechanisms of the mind. So the question from what I've told you, the question that we want to ask is, can we know the thing in itself, das Ding an sich? And the answer, as you might expect from any sort of philosophical disquisition, is nuanced. Yes, well, first of all, we cannot know the wavelength composition of light coming from a surface unless we are armed with a very, very powerful spectrophotometer. And when you go to Regent's Park, you don't know the wavelength composition of light reflected from different surfaces in these different lighting conditions, but you know the color. On the other hand, what you do know about the thing in itself, but you may not say so unless you know physiology and anatomy and color theory. What you do know is that different surfaces have got different reflectances for different wavelengths of light. So when you look at a green surface, you know that green surface is capable of reflecting more middle wave light than the surrounding surfaces, and therefore it is green. And a red surface capable of reflecting has a higher efficiency for reflecting red light than surrounding surfaces, and therefore it is red. And that knowledge is, in fact, the knowledge that you translate into color. How that translation is made is not known, but just like taking the letters of the alphabet and translating them into words. So let me come to conclusions. Conclusion 1, light as electromagnetic radiation has no color. Colors are constructed by the brain through a ratio-taking mechanism. Area V4 of the cerebral cortex is central to that ratio-taking mechanism and the construction of color, by which I do not mean that it acts in isolation. Conclusion 2, through the construction of colors by a ratio-taking mechanism that is dependent upon the reflectance of surfaces for light of any given waveband, the brain makes itself independent of the constant changes in wavelength composition of light reflected from surfaces and thus stabilizes the world of color. Conclusion 3, color is therefore a language given by the brain to certain unchanging properties of the world outside, namely the reflectance of adjacent surfaces for light of the same waveband. Through the language of color, the brain can acquire knowledge about the thing in itself, das Ding an sich. That knowledge is about the reflectance of surfaces for different wavebands, and the color is a shorthand notation for it. And finally, reflection of the conclusions from more general nature, which is what I have taken away from my colorful experience in the world of color. First of all, to beware of authority, I relied in a very unhealthy way for too long on the authority of people who knew a lot about color, but did not know about color mechanisms in the cerebral cortex. Therefore, well meant though their advice was, I should have looked for alternative ways of doing things. Secondly, that I am an academic, and I think the academic world is the best world in the world. It's hospitable to new ideas. It's full of intelligent and able people, but it also has a very dark side in which you can try to stop people from proceeding with the work because it disagrees with their views. And thirdly, to beware of experience and the power of experience. Now, I think that I would like to, if I'm given 2 minutes, to quote from two writers. The first one is a man who said that it's not true that we don't make the same mistake over and over again. We keep on making the same mistakes, and then we call it experience. This is the man who wrote, before most scientists understood it, the following lines. We now know that we do not see with the eyes or hear with the ears. These are really channels for the transmission, adequate or inadequate, of sense impressions. It is in the brain that the poppy is red, that the apple is odorous, and the skylark sings. That man was Oscar Wilde, and he wrote this in De Profundis, his last work, which is a long letter. And finally, I would like to end with the quote from another great artist who knew about science, and that is T.S. Eliot, about what he said about experience. It seems to us that there is only a limited value in the knowledge derived from experience. The knowledge imposes a pattern and falsifies, for the pattern is new in every moment, and every moment is a new and shocking valuation of all we have been. Thank you very much.
Thank you, Professor Zeki. That was a fascinating talk. When we think that the remit of the Paton Lecture is to promote interest in the history of scientific experiments and ideas, I think you really hit the nail on the head there. And in particular, I mean, I obviously have a great interest in the history of science, that I find talks of this type particularly interesting and particularly insightful when experts such as yourself, who have worked in the field for decades, can reflect on that field as it was when they came into the field, what their particular input was into the field, and how that field and perceptions about that field have altered over time. And I think there's a very important lesson to be learned, as you spelled out towards the end, that researchers must be willing to evolve in their field. They cannot continue on with the same ideas through decades of research. And just to sort of finish off with a brief historical little anecdote, that I was very pleased to hear you talking about Sir Isaac Newton, because just last week I was on a bike ride and I went past Woolsthorpe Manor, which was obviously where Newton was born and where he did some of his key experiments on light and optics. So thank you very much for that talk. That was fascinating. And I think we will draw this session to a close. Again, on behalf of the Physiological Society, thank you very much, Professor Zeki.
Thank you.